Welcome to our channel by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is artificial fertilization. You are aware about artificial fertilization. It is the crossing between two different varieties with desirable characters to obtain third variety with both the characters or desired characters. Improved variety with desired character is known as artificial hybridization. It can occur naturally also, but it takes time or it is very rare. It takes thousands of years to have a origin of new variety with natural crossing or hybridization. But artificial hybridization, it make it possible within one generation that we can obtain a new variety with desired characters. So let us learn as a part of syllabus that what is artificial hybridization. So it is the crossing between two different varieties of a crop or varieties of a plant or varieties, two different varieties with desired characters. Say there are two varieties, for example a Kesar mango with sweet taste and very attractive saffron color to the juice, and Rajapuri mango with a large size. When we have to bring these two together by artificial hybridization to have the third variety with improved quality, that is having large size of Rajapuri with Kesar taste. It is possible by transferring pollen grain of either of any one, either Rajapuri or Kesar and vice versa, to the stigma of the flower. So how artificial hybridization is done, let us understand first and then the process. Here there are two flowers. We can take these two flowers with this stigma, style and ovary. This is a carpel, this is a female reproductive organ, and here these are the anthers. These are the anthers, male reproductive part. Here these two flowers we have selected for two mangoes, one Kesar mango with sweet taste and Rajapuri mango for its size. Here sweet taste and here large size. We have to bring these two characters together, this sweet taste and this large size, so as to have a mango with sweet taste plus large size. So what to do then? How it has to be? Either to transfer these pollen grain from this flower to stigma of the Kesar mango, or pollen grain of Kesar mango to the stigma of the Rajapuri. This is called crossing. Now it is the practice to develop the improved varieties in plants, and the process is to avoid the cell pollination, very important, because we have not supposed to produce only Kesar or Rajapuri, we have to produce the combination. To avoid the cell pollination, that pollen grain from anther of this flower to the stigma or pollen grain from anther of this flower to the stigma has to be avoided. cell pollination to be avoided. To avoid the cell pollination before maturity, we have to cut the anther so that pollen grain from Kesar mango will be transferred to the stigma of the Rajapuri mango. This process is known as emasculation. So, what are the steps in artificial fertilization? So, one, that is selection of desired character. This is first, desired character, which supposed to be together in improved varieties. Therefore, first step is selection of plants with desired character. Selection of desired character or selection of plant with desired character. Second point is cutting of anther before maturity to avoid the cell pollination. That is called emasculation. Now, in which flower that emasculation need to be done? In bisexual flower, which is supposed to be cross-pollinated. In unisexual flower is not needed. So, here emasculation is the cutting of flower, the cutting of anther, cutting or removal of anther before maturity, to avoid the self pollination. To avoid the self pollination is known as emasculation. And in which flower it is needed? It is needed in bisexual flower having male and female together. In unisexual flower it need not. Okay. Now third, after emasculation, when that anthers are cut, till maturity, this stigma should not be contaminated by any other pollen grain than desired pollen grain. So, it has to be bagged. That next step is bagging. How to do bagging? To understand bagging is here a small flower to show you to understand that bagging. This is stigma, style and ovary. So, now here it has these anthers are cut down. These anthers are cut down, and now that till to that maturity, this flower on that plant has to be bagged. So, it is tied up. Now, there will not be transformation of anther when stigma get matured. Emasculation is done and bagging is done. To avoid, very important, it is to avoid contamination by any other non-desired pollen grain. I hope you got it. What is that bagging? Now, when that flower get matured, stigma become receptive, then this bagging to be removed and dusting of pollen grain from Kesar mango to the stigma of the, so dusting of stigma, receptive stigma with desired pollen grain or pollen grain from desired plant with desired character. So, this is next step, dusting, very important. Bagging, emasculation, selection, these are the key words. So, after dusting once again to bag, because see what to do after dusting again. So, flower, small flower to understand. This is stigma. To show the dusting of pollen grain. Here pollen grains are dusted. And now flower is ready for fertilization. So it has to be rebagged to avoid the contamination, because another pollen grain may fall and germinate before that, to avoid that pollination. So these are the pollen grains from desired character, and here this is a rebagging and tie-up. There are holes from downside to have the air. So fifth step is rebagging till the fruits and seeds, or seeds in fruit and seed are developed or produced. So these are the 5 steps to be remembered as a sequence, and question can be asked. So very important to know. First step is selection of desired character, second is emasculation, third is bagging, fourth is dusting of pollen grain of the with desired character and rebagging till that flower get such fertilization occur, fruit and seeds are developed. Now we will obtain finally that what we obtain is a large size mango, with desired characteristics. So here this is a mango, large size mango with Kesar test. So finally we obtain this mango having both the character, size is large and Kesar test. So finally this hybridization is completed. So what we learn? Artificial fertilization. What is the process of artificial fertilization? Is selection of desired character. That 2 plants are selected with that desired characters. Now that plants which are using at when they begin to flower, so to avoid the cell pollination in bisexual flower, anthers are cut down before maturity to avoid the cell pollination is called emasculation. After cutting of that anther before maturity, this flower has to be bagged to avoid the contamination, because pollen grains from other plants may get transferred to the stigma by the time when it becomes receptive and contamination may occur. So to prevent the contamination, that bagging has to be done. After maturity, when stigma become receptive, that pollen grain from desired, that is from Kesar mango, to be dusted on the stigma of the flower of Rajapuri mango so as to get fertilization and have that character. So after dusting, once again has to be rebagged, because by the time when we have dusted pollen grain, they may get fly away with wind, and other pollen grain from non-desired or maybe from Rajapuri itself sit on that stigma and germinate and produce the mango that would be Rajapuri. So to avoid that cell pollination, to avoid contamination, this has to be rebagged. So rebagging till that fertilization is completed and flowers, they shed down their petal and small fruit begin to produce with seed. Then that bag can be removed, fruit can be obtained with both the character, that is large size like Rajapuri and large size like Rajapuri and taste like Kesar. So here the fertilization is completed. I hope you did understand what is artificial fertilization. Question can be asked, write short note on artificial fertilization, or in NEET question can be asked, arrange the sequence, arrange all these events according to the sequence of process of artificial fertilization. This can be reshuffled. Dusting can be given before, emasculation can be given later, and that what we have to arrange that according to the sequence of process of artificial fertilization. And question can be discussed. NEET, the question which supposed to be asked in NEET can be discussed in second part of videos where we prepare for NEET examination. That includes question based on NCERT books. Second part will be question on thinking based or conceptual. Third, that question which asked in NEET till that NEET examinations are conducted. So these 3 parts, MCQs in 3 parts will be discussed as a separate series of video, and that very soon we are going to start. As you can see that given on NET, or you can see in Google that these diagrams are given here. See this cutting, the way that anthers are cut before maturity to avoid the cell pollination, and the process which shown here, that bagging and rebagging. So how to bag and rebag, it is shown here. I hope that you can go through so that you can match this diagrammatic information given on net with the explanation given by me, and just to update your knowledge. I hope you did understand entire process of this artificial hybridization explained by me with the sequence of that process. Even if you are in doubt, you can ask in comment section, and I appeal you to like and subscribe our channel, as well as press bell icon to receive new videos as and when they are uploaded. Thank you.